All right, so the first part may be too theoretical, too new. All right, maybe after this we can have a discussion if you have extra time. If you have questions, this and that, I'll walk back and forth. So I'm here. So as your dean said, take advantage of it. Okay, so the next part for crystallography is mostly application or as a user of crystal structures or structures in general, what tools are available. Some of this you may be familiar with, like PDB, you're all familiar with, which is good. And then maybe others as well. So, so this slide is all on crystallography program, which I'll skip because that's maybe not of interest to you unless you're really doing crystallography yourself. Otherwise, this probably doesn't matter too much. But this is probably more useful for structural analysis. And that these things I use quite a bit myself, looking at structures. So I'll go through these one by one to sort of show you some of this you may be familiar with. If you are, great. If not, I'll explain what they do. Okay? So PDB, you all know. So I won't spend much time talking about it. So this already explained. Okay? Resolution is somewhere here. So you can sort of, for each structure in the PDB, you can sort of see the resolution of that structure. And what you may not have heard is a program, a server called PDB Redo. Okay? So as I mentioned, our structure from 2006 will be all the way over here. So what PDB Redo did is it took the deposition, structure factors, and the model, and automatically re-refined the structure using modern technology. So then the structure will be somewhere in the middle, or hopefully maybe even better on this side. Hopefully that will improve the quality of the model if you want to look at. Okay, so that's sort of something I use once in a while, not very often, but I'll show you one example later today how PDB Redo can be useful. And Molprobity, another server that you can use down at Duke University. So here what you do is you upload your protein structure and it tells you the quality of the geometry. Geometry being bond length, bond angle, torsion angles, Ramachandran plot, all that stuff. It tells you the quality of your structure. So green is good, yellow means warning, red of course is not good. So here, per rotamer, sidechain rotamers, which are new to you, so I will make sure to explain that a little bit later. It's better to show that actually using actual graphics so you can see the difference in the sidechains. And then Ramachandran outliers, so the goal for Molprobity is less than 0.05%. So I have this structure, which is one of our structure, 0.2%, which is yellow. Didn't kill us yet, so which means it has two outliers in this case. You have to go in and tweak your model to fix that problem. So all of these basically give you a sense of the quality of the structure. And this information essentially summarized in the PDB geometry thing, in the little plot right here, sorry. Right here. Right here. Okay. And then another server that's quite useful, very powerful as well, it's called Conserve. Have you heard of it? No? Okay. So Conserve means conserved surface. So what this does is it takes your PDB file, you upload the PDB file, which is to the server, which is based in Israel. And then it'll extract the sequence of your protein based on the PDB file. And then it does a blast search against all sequences, find all its homologs. And then it calculates conservation at each residue of your protein. And that conservation then can then be used to color the structure. Okay, so the typical Conserve convention is red, or whatever color that is, it means conserved. Blue means variable. White means somewhere in the middle. So by doing this analysis, you can readily find the region, for example, for this enzyme or for this protein here, a large area that's highly conserved. Also, this shown at the surface, you can see that in the middle is a very deep pocket, right? So it's a deep pocket, highly conserved, and it's an enzyme. So if you're looking for the active site of the enzyme, where would it be? It will be right here. So this is another way to sort of help you look for things, how well everything is conserved in your structure. So while away from the active site, things are very, very flexible, or variable, sorry, not flexible, not conserved. So another very useful tool to help you locate the important parts of your protein. The important parts typically are all quite conserved, so this will help you to look for those. And then another thing is, if you have a protein structure, you wonder what other structures look like your structure in 3D. Because structure is more conserved than sequence. So things that may not be homologous in sequence, but may be similar in structure. So to look for structural homologs, there are many tools available. The one I use a lot is something called DALI, which is a server based in Helsinki, Finland. So again, you upload your PDB file to the server, and then it'll come back with structural homologs. Okay? More interestingly, you can search against the PDB, or you can search against AlphaFold DB. You know what that means? That means you can search against all the predicted structures as well. Now, you don't have to be limited by only those you have done the structure of, or somebody done the structure of. You can search against almost all the structure available. And in this AlphaFold DB search, you search against all the human proteins, all the Drosophila proteins, all the yeast proteins, all the bacterial proteins. Well, based on organism, you have to search against a specific organism. But you can search against all. So we often work on human proteins, so I search against all human proteins, see whether there are homologs or structural homologs of my protein. Okay? And the results is a list like this. So for your protein, the homologs are listed 1 through, just go down, depending on how many homologs there are. They're sorted by something called the Z-score, which a measure of the similarity between your structure and this structure in the PDB. And this is based on PDB entry code, chain A. So the entry code followed by the chain in that entry. And then it gives you RMS distance between your structure and this structure that's in the PDB. So 2.5 angstrom in C alphas only. Okay. So that's actually pretty similar structures. And then it also give you the sequence identity between your structure and this homolog in PDB. Only about 17%. Very low sequence conservation, but fairly good structural conservation. So that's why 3D homolog searches are actually more powerful to finding homologs. So that's why we use this quite a bit. Okay? And then AlphaFold, of course, you're all familiar with, or at least, well, no. Some of you heard of it. But anyway, so AlphaFold, as I said, 2021, the paper came out in Nature. So they have a website. You can go download your structures. But this website actually not very, you have to put in your name or your protein. And that oftentimes is a search routine not as good. So I actually rarely use this anymore. So I'll show you another way of finding AlphaFold structures, much easier way of doing it. But anyway, when you do find your homolog, it shows you the predicted model right here, colored by AlphaFold color scheme. So basically, confidence, how confident you are in the prediction. So blue is very high confidence. Cyan is good confidence. And then yellow is okay, maybe, maybe not. And then orange is, don't even think about it. Okay? So you can sort of see that this structure, something has a domain right here, alpha solenoid. Do you know what that is? Alpha solenoid, alpha helical repeats. So that's predicted pretty nicely. And another alpha solenoid right here, you can sort of see this one corresponds to this blob. This one corresponds to this blob here. And then these two blobs are not talking to each other. They are flopping around. Two domains flopping around. Don't do anything. There's a little domain down here that talks to this domain a little bit, but doesn't talk to this domain at all. So there's a lot of information you see from looking at a prediction like this. But anyway, so this is sort of how you look at AlphaFold models. And then, as I mentioned, I'll show you a couple examples. So this is one example. So here is a structure that we determined by selenomethionine crystallography. So that's shown in yellow. And then when we determine that structure, there is no model. Nothing of the structure has been in the PDB. So there's no bias to AlphaFold or Rosetta fold. And at that time, AlphaFold, you cannot put in any sequence and predict for you, but Rosetta fold can. So I put in this sequence into Rosetta fold and ask Rosetta fold to predict the model for me. So now I have the independently predicted structure and then experimental determined structure of the same protein. So I can compare them. Okay? So green is the model from Rosetta fold, and yellow is our actual crystal structure. You can see overall they are actually remarkably similar. RMSD 1.3 angstrom. And you can see the beta sheet here almost right on top of each other, showing the quality of the prediction. But then you can also see the alpha helices are somewhat different, and this beta hairpin is also somewhat different as well. And this might be crystal packing, we don't know for sure, but some of these are probably real differences between the actual structure and the model structure. So it's a very good quality prediction. It tells you exactly what the structure looks like, but then the details probably is not quite there. That's why RMSD still is 1.3. This is only C alphas. If you include everything, the side chains, everything, it would be even more different. Okay. So this is 2021. I mean, when this first came out, obviously. Okay. Don't worry about that. So another case, remember molecular replacement using AlphaFold models? So here's a case. So let me tell you the gory history behind this case. So we grew crystals, we being my student, I didn't do anything. My student grew crystals of this protein. And we went to the synchrotron in November 2011, so now 14 years ago now. We collected data to 4.3 angstrom resolution. You say, that's horrible. Yeah, it is, but that's the best crystal we have. So that's the best we could do on that crystal. We took quite a few. We tried all of them. The best we get is 4.3 angstroms. So we need to solve the structure. It's a human protein, very important protein. We'd like to get the structure of this. However, somehow, we could not repeat the expression. We cannot make the protein. It's expressed in E. coli. Somehow, we cannot reproduce that expression. And of course, without that, we cannot grow the crystal again. And then, without that, we cannot improve the crystal or solve the structure. And we cannot make selenomet because we cannot make the protein. And then we say, okay, we know the sequence of the protein. Can we find the homolog? No homolog in PDB back then, 2011, 2012, 2013. I think we struggled for a couple of years. No sequence homolog, so we don't have a model for molecular replacement. So because of this difficulty, and especially we can't even express the protein again, we thought maybe the crystal is actually a contaminant from E. coli. We purified something that's not really our protein. We grew the crystal, and then when we next time tried, we didn't purify that protein, and then that's why we couldn't solve the structure, couldn't reproduce it. So here comes 2021. So AlphaFold came out and said, okay, let's see whether AlphaFold is good enough for me. So I took the AlphaFold model, put it into molecular replacement, boom, structure came out. Okay? And that's what the crystal looks like. Essentially, this little yellow piece here is a unique molecule in the crystal, and everything else is crystal pack related by symmetry of the crystal packing. So essentially, this crystal is about 85% solvent. That's why it differs only to 4.3 angstrom resolution. It's very poor quality because it's mostly water. But that's the best we can do. So now, 2021, 10 years later, we got the structure. Can we publish it? Probably not. This is only 4.3. But still, it shows that essentially the power of this model that basically will be a starting point for you to determine almost any structure you want to work on. If you don't have any other experimental structure, start with AlphaFold or Rosetta Fold, and then you probably will get there. Okay, so the power definitely is there. Okay, so now AlphaFold 3 came out last year. So, and AlphaFold 3 is an improvement over AlphaFold 2, the one that came out in 2021, because now you can predict protein-protein complexes, protein RNA, DNA complexes, and protein small molecule complexes. Almost anything you want to work on, it can predict for you. Right or wrong, that's your choice, your decision to make, but you can certainly make a model for you. Okay? So, and most importantly, actually, now they have a server that you can use. You can go there and run through Google. Very powerful server. I use it all the time. And very simple input interface. You can put in 5,000 residues maximum. You can put in a very large complex with 5,000 residues into it. It'll do the calculation for you. But then it has a limitation in the sense that you can only run 20 jobs a day, which is still a lot. I've never run over that limit. I've done 15 a day, I think maximum once. So, and 5,000 seems to be a lot. Actually, something we work on, I'll show you later this week. 5,000 is still not enough. It's a megadalton complex, but still, it's a very good starting point. So essentially, it's a very useful tool and also very, very fast. Even if you put in 5,000 residues, it runs over 20 or 30 minutes, and then it gives you the model. I guess maybe I'll show you that as well, or maybe Professor Shukla will show you how to run this. Very powerful. I mean, we can do it at home, on the home computer as well, using GPUs and all that stuff. It'll probably take a day or two. So, and then last time I tried to do something like this at Columbia, now there were a brand new GPU workstation with 4 GPUs. It basically drew so much power, it tripped the circuit breaker. So we lost power. Anyway. Okay, so that's AlphaFold. And the next thing I'll tell you about is something called UniProt. You probably heard, I see you nodding. Yeah, so you have heard, anybody else heard of UniProt? Yeah, okay. Yeah, great. Very powerful, very useful database. I use it all the time. Any protein I want to work on, somebody tell me, okay, this is a protein you should look at. It's okay, I'll look at UniProt first. So you put in basically you put in the name of the protein, it'll find it for you, you click on it. This is actually the old interface, sorry. But anyway, it gives you a lot of information about this. And then on the left, there's something called structure right here. So that structure will give you all the structural information on this specific protein, including the AlphaFold model in there. So this is now how I find AlphaFold model for my protein. I go to UniProt, I look at all the other structures done by NMR, crystallography, or EM. They're listed first. At the very bottom is AlphaFold model. And so that's where I download it and look at it if you want to. Sometimes your protein has no experimental structure, only the AlphaFold model, and that will be there. You can look at it. Together with many other interesting, useful information. And I will, well, I may or may not get there, but anyway. So maybe let me tell you the story here since we have a little bit of time. So we showed 2 proteins from a complex, okay, in humans. And then we know that one of the proteins is a homolog of another protein that I will tell you a lot about later. So I wonder whether that protein has a homolog as well. And there's no knowledge about that protein has a binding partner just like these 2 proteins. So the way I did it is go into UniProt, there's a protein complex tab right there. There's a database is reference to. It's something called a BioGrid. BioGrid is actually I'm on the network, so I can go there. Okay, let's see. Where's my, here. Here. Okay. So you type your, this is better? Okay. Well, let's try it. So you type in the name of your protein, which in this case CPS23, and I want the human one, so let's go to the human one. And then it gives you the tabs right here and all the information about this protein, what's known about the protein. And then, so just scroll down. It tells you the protein is nuclear, which most times is correct, sometimes not. Disease variants, and PTM, post-translational modifications, and then interaction. So what does protein interact with other proteins? What are the binding partners? So, and then the databases. So you can see database right here is something called BioGrid. So these are mostly done using omics, mass spec, pull downs, all that stuff. So omics find the partner, it goes in the database, it's reported. Whether it's real or not, don't know. So the protein I was looking for is CPS23. Whether CPS23 has a binding partner just like the other two proteins. So what I did is just go into this database, BioGrid. Okay, so which CPS23 is all called CPS3. You'll know that soon. So now it's just a list of all the binding partners. Some of these I know because I work like CPS2. Well, so that's a binding partner of CPS3. Well, that's why they are called CPSFs. Some of these I do not know. So the way I show whether they are complex or not, I use AlphaFold to predict the complex. See whether the AlphaFold can predict the complex for me. If it cannot, I say forget it, move on to the next one. So I manually screen through this list right here, one by one. And I found one. And that protein is this one right here, UBE3D, which now is pretty high up because since I found it, many people publish on that. So now this is updated all the time. So when I was doing it, this is way down the list. So this was a number 19. I tried 19 different combinations. And number 19 is UBE3D that gave me a complex. It turned out to be actually the one we were looking for. Now, actually, it's pretty high up now because it's a real complex. So now it has many other evidence now, so it goes up. So these are sorted by number of evidence right here. You probably cannot see in the back, but anyway, the number of evidence that they do from a complex. So AlphaFold can be very powerful in that sense as well. So you have a hypothesis that your protein has a binding partner, but you don't know where it is. You can go to UniProt and then go into the BioGrid or any other database you like. I mean, BioGrid seems to happen to be the one I found first. It could be any other database you have, and then you can just go through them either manually or you can write the script if you're good at Python, other things, and then just go through one by one and then look for it. And you may get lucky. Or may not. But anyway, at least you can try it. You have a way to go there to figure it out. Okay. We're almost done. So the last one I'll show you is something called Brenda, which is an enzyme database. I think many of you here work on enzymes. So this is a very useful database that has a lot of information about any enzyme you want to work on. So I go here quite often as well because we work on enzymes quite a bit as well. So it gives you KIs, IC50s, inhibitors, all that stuff, KMs, all that stuff are in here. Okay, so something you can look at as well. So yeah, one thing I forgot to mention here on UniProt. So I showed you had the structure tab. You can go there, find all the structure for your protein. However, keep in mind, that's only for that specific protein. For example, if you look at the human protein, but there's a structure for the mouse protein, you will not see it in the human entry. Okay? So keep that in mind. So it doesn't mean that you don't see in the human entry, doesn't mean there is no structure for the mammalian species. It could be one for mouse. You will see it in the mouse protein entry, but you will not see the human protein entry. How do you solve that problem? You blast. So you take a protein, put into BLAST, you select in BLAST PDB as a database, and you find all the structures that are homologs of your protein based on sequence in the PDB. You know how to do that, right? Okay, you can select database when you do BLAST. So select PDB as a database. Now you find all the structures that are homologs to you. Okay. So I think that's it for crystallography. And what time is it? We have half hour. So we can go through discussion now, or I can go to EM next. Tell you a little bit about Cryo-EM. Any preference? EM first? Discussion? Okay. Let's start. Go ahead. Questions? Yeah, okay, I'll walk back. I can hear you. Sorry, I guess. Yeah, go ahead. How can you tell the difference?
How can we select the best template for our given sequences for the structure prediction in AlphaFold?
Okay, so you have a structure of AlphaFold and you have?
We have the sequences and now we want to predict our structure.
You want to do what? I'm sorry?
I have a sequence and now I want to predict the structure with the help of AlphaFold. So how can we select the best template for that in AlphaFold?
You have a sequence. You want to use AlphaFold.
Yes.
Just use the server I showed you. So
it gives lots of templates for our sequences on that they have that structure is going to predict. It gives us lots of templates.
different templates?
Yes, sir.
Templates for?
For that our structure is going to predict on the basis of that template.
I'm not sure I fully understand. So you have different templates of
Any structure, AlphaFold suggests that different templates on which it will suggest our structure.
I'm not sure I fully understand. So you have one sequence, right?
Yes, I have one sequence. Now I want to know what our structure looks like. So for that we have to select the template in AlphaFold, no?
No, you don't put in a template. I'm sorry, I don't understand. So you need the structure template.
Yes, sir.
No. AlphaFold takes only the sequence.
Okay.
Only the sequence. So I'll show you the server, right? So let me show you the server. I'm sorry, I guess I'm in your way all the time. Here. So you can just put in the sequence right here. So you tell it it's a protein. This is drop down right here. So you tell it it's a protein or nucleic acids. You click on this, it's a drop down menu. Small molecule, protein, nucleic acid, RNA or DNA, or metal ion. You can do metal ion as well, magnesium, zinc. So this is drop down, how many copies you have, and then here you put in the sequence. That's it. That's all you do. And then if you have some molecule, right here, it'll be a drop down menu. You can select a few small molecules, limited number of small molecules. So the server is limited in terms of small molecules, but if you have AlphaFold 3 installed on your own machine, you can use any small molecule in the calculation. But the server, there's a limited number. Let's just go there. Sorry. Here I have one. So here's the AlphaFold server. That's just Google it, you'll find it. So you can tell it's a protein or, hold on. Here you go. So you can tell it whether it's protein, DNA, RNA, ligand, iron. And then if it's protein, just give it the protein sequence. It doesn't have to be the full length. It could be a domain. It could be a smaller piece. And that's it. That's all you put in. And all you can put in the RNA sequence. Or you can put in mixtures, everything. And then when you're ready, just say go, and then it will calculate for you. So it's a very powerful thing. I'm sure it's very easily accessible. I don't know, have you anybody tried it? The AlphaFold, you have tried it? Did it work for you? Huh?
No, sir.
Yeah, very useful. I mean, we are structural biologists. We use it all the time.
So it works only when there are homologous sequences available. Or when we can work for any new sequence as well.
Yeah, anything. Anything.
So we do not require any homologous sequences.
No, it's any sequence you put in. I mean, of course, essentially in the end is what you get out of it. So let me show you what you get out of it.
Like you said, that Rosetta Fold can predict the structure de novo also. But AlphaFold cannot do that.
Well, AlphaFold 3 is more powerful in many ways. Yeah. So this is based on
What is
AlphaFold.
the difference between AlphaFold 3 and Rosetta Fold?
Mm-hmm.
So what is the difference? What software can we use?
In what?
Which is more preferable, Rosetta Fold or AlphaFold?
Oh, Rosetta Fold. Yeah. So Rosetta Fold has essentially very similar capabilities, let's say, but doesn't have a server in the sense. So we have Rosetta Fold installed on our GPU machine in-house, so I can run it. So I can get.
So we can run it online.
Yeah. And now AlphaFold you can run on the server, but as I mentioned, for a small molecule, okay, let's try it. So if you have a small molecule, so here you have an example, I put in RNA here. So this is the RNA sequence. That says RNA. It's RNA sequence. So let's say we have another thing, add entry, and we say small molecule, ligand. Now you can see this is the drop-down menu. So you click on that. And then it gives you choices, ADP, ATP, the common CoA. I don't think CoA is GTP, FAD. It has a good number. Some of you work on plants, chlorophyll's there too. So it has a good number, but not, I mean, of course, not everything. Small molecules are millions of them. So if you use a server, these are the only ones available for you. But then if you install AlphaFold 3 on your own GPU, then you can use almost any small molecule. But the server is only limited to these. So essentially, you can put everything in, and then what you get, so let me see, which one should I show? See how many I've done? This is my real work, actually. So here's a prediction from AlphaFold for basically quite a few different proteins. And I said maximum is 5,000. This one I got to work with 5,058. I don't know how I succeeded, but anyway, it worked. So this prediction has 5,058 residues in it, protein residues. So a big complex of many different things. Color, same way. Blue is good. Cyan is reasonable. Yellow. And then orange is bad. And so that's another warning for you using AlphaFold 3. You got to look at this coloring scheme because AlphaFold 3 for really bad regions have a strong tendency of making them into alpha helices. So you can sort of see these really beautiful alpha helical regions right here. You can sort of see, right? Beautiful domain, a domain of alpha helices, but that's not real. So AlphaFold will make them into a spaghetti. Loops going all over the place. AlphaFold 3 make them into a helical domain. So you have to ignore them because they are not really there. But even if you ignore them, the rest actually is a lot of information there that can help you design better experiments, help you get a better understanding what this protein complex looks like. Okay, so, and if you're looking at protein ligand interactions, you can put in a ligand and see how that interacts as well. And there, the confidence is probably not as good as protein-protein or protein-RNA interactions. But anyway, but at least give you some ideas. Okay, so any other? Oh, okay, so PyMOL, are you all familiar with PyMOL? Okay, so what you do is you can download right here. So let me do that.
Sir,
Yeah.
and what parameters these sequences are predicted? How can experience
Yeah. So these are based on something called PLDGT, color based on PLDGT, but there are many other values that come out. So there's also IPTM. It's an interface PTM. So that number should be high as well. So there are many parameters come out, and more importantly, look, this matrix, so-called predicted aligned error. So you can see error here, 0 is very green, white is very, very, very bad. 0 angstrom error, 30 angstrom error. So you want things that are darker colored. So you can see that, so this is a complex, several different proteins, how many there are. Plant protein, 1, 2, 3, 4, 5, 6 proteins. So these are 6 different proteins. You can see there's some contact in this protein and that protein because off diagonal is the interaction. And then within this protein, a lot of interaction. But then this part, there's really not much interaction. So this plot also tells you whether there's anything important going on. For example, there's a stripe right here that's interacting. So you have to look at not only this model, also look at this together. All the information together will give you a sense how believable your structure, your prediction is. Okay? And then you can download the structure. So let me. Is this useful? If not, we can skip on. Is that helpful? Yes. All right. Wow. Your network is very slow, so I guess we'll wait for that. So while we wait for that, any other questions? Yeah.
Sir, wonderful talk.
Thank you.
The thing is, like, I had the opportunity to work on a protein that has a huge number of disordered regions. And when I'm putting that sequence into AlphaFold, it is giving a very low PLDT score, that's the yellow region. And I used different algorithms. Beside AlphaFold, I used Bold's one, that's from the MIT. And also recently one algorithm came up that is Proteinics recent server. So different, like in case of AlphaFold, there is a huge region that is in disallowed from a form. But when I'm using the different algorithm, it's giving a different structure. Also, the disordered region is structurized in that different algorithm. Also, the disallowed region is very less. So, the different structure is coming up from different algorithms. Which one is correct? Which one is not?
Very good question. The question is, you run different prediction programs, you get different answers. Solution is correct. Well, that's why we need structural biology. It's perfect setup, thank you. So that's why you actually do a structure. Or maybe they're all correct depending on what condition you're at. So it could be flexible here, but then when it's the complex stabilized this way, once a different complex stabilized different way. That could be possible as well. All different conditions, it could have different effect on the conformation of the protein as well. So it's actually quite often we see these so-called IDR, intrinsic disorder regions. They are disordered on their own, but then when they are in the protein-protein complex, they could become stabilized. So, and that's something that you find Used to be, I mean, we have quite a few examples we find by chance. We don't even know that they are ordered until we did the structure and say, oh, where did that come from? Came from a disordered loop that was basically ignored, but then that came out to be actually the part that's important for the interaction.
In
Now,
the protein, there is two domains that is connected by a disordered region.
right,
That disordered region is supposed to be work on DNA binding.
right. So the disordered region binds DNA, that's the idea? Or, so can you see that by any biochemical assays or anything?
No, sir, we haven't did that, but this is predicted. Like,
So, yeah. So again, you need experimental data. In my mind, I'm an experimentalist, so I will say you try to get some experimental data. And then in this case, maybe you can try put into AlphaFold 3 and see whether you can predict the complex between your protein, that linker region, and the DNA, whether it makes sense or not. I guess once you have a model, if you feel that's real, the next thing to do may be mutagenesis or binding assays, all that stuff. You can try to assess. And that's something we do as well. Oftentimes, we cannot get a structure, even though I think we're pretty good at it, but we can't get everything we want. So we have to essentially sometimes predict the structure and then followed by mutagenesis, purification, gel filtration, pull downs, everything else to assess the model from AlphaFold, see whether it makes sense or not. Oftentimes, the story comes together, and we have certainly published papers where we show AlphaFold model and biochemistry together. So that the example I just showed you, sorry. The example I just showed you, we haven't got, well, by the time the paper got published, we don't have an actual structure yet. So we published the AlphaFold model. But since then, we have determined the structure using Cryo-EM. And overall, big picture is the same, but there's a little wrinkle as well from the actual structure, which is good. So that means we have a better story now to tell on the actual structure. So these are, I think they are mutually helping each other. Having the model give you new insight, new ideas to try, and having the structure improves on that as well. Does that sort of?
yes, sir.
Okay.
Thank you, sir.
Are you doing experiments also, or doing mostly calculation?
Yes, sir, I'm doing little bit computational structural biology, bioinformatics, and also few wet labs.
Okay. All right, that's done. So let me show you this. The download is done. So let me show you the. So essentially, it produces 5 models. So the 0, 1, 2, 3, 4. So 5 models. It's 5 different neural network models. And then so each one produce a model. They are sorted based on confidence. And these are CIF files, CIF, rather than PDB. PDB is old news. I guess you know that, right? So the new format, CIF in crystallographic information format, CIF. So these are CIF format files, but doesn't matter. PyMOL opens just as well. So we can open the PyMOL. So here is the entire model. And then, I don't have the mouse, it's okay. So you can color by chain, and then it shows you 6 different chains. So can you see okay in the back? Yeah? Okay. So it gives you all the structures. But then this is everything that came from AlphaFold. As I showed you earlier, a lot of that is actually not believable. So for that, you can color. So the confidence from AlphaFold is encoded in the temperature factor field of the PDB file. So you can color by temperature factor. So unfortunately, in AlphaFold, good confidence means 100%, remember? Which means very high B-value. So if you color by B-value, it's red. Which means when you color by B-value, red parts are the good part. And blue part is the bad part. It's the inverse of the AlphaFold color scheme. But anyway, so You can sort of see now, essentially, you have, remember those two alpha helical domains that are low confidence? They're cyan here. So essentially, things that are blue or cyanish, you don't trust. You don't want them, essentially. And you can, you're in PyMOL, you can hide them. You can hide everything, B less than 50. They're all gone. So now you look at only the part that has good confidence, and then you can analyze your structure and then get insights based on that. So you're not biased by the part that are low confidence. Okay. And then, as I said, there are 5 models. So what you should do is look at all 5 and make sure that they are similar to each other. So overlay them together and then see that whether they are really different. If they're very, very different, all 5 are different from each other, that means probably you don't have the right answer. If they're all similar to each other, probably you have the right answer, or probably you had the same wrong answer. But anyway, at least it's better than all 5 being different. When that happens, probably very likely AlphaFold doesn't know what to do with it. It does happen. Especially 2 proteins don't interact. When you make a prediction, you get all kinds of different things. That will be a good control. Okay. Any other questions? Okay. So we have 5 minutes. What do I do? So should we finish early, 5 minutes, or want me to go into EM? Huh? Start with EM? Yeah. Okay, all right. Maybe that's all I can do. Okay. So we are here talking about EM owing a lot to these 3 giants in the field, Jacques Dubrochet, Joachim Frank, and Richard Henderson. And I understand, I heard this morning that Joachim Frank was here in 2017, right after he won the Nobel Prize, is that right? Okay, so he obviously at Columbia as well. I have him teach a guest lecture to my students once a year. So I have borrowed a few slides from him as well. So maybe I've seen them already, but anyway. Okay, so modern EM are driven by these giant boxes. Sorry. So on the left is a 200 kV microscope, or glaciers, that mostly we use for screening the quality of a EM. Right now, it probably doesn't mean anything, but soon, hopefully, it'll mean something. We'll use it for screening the quality of our grids. And also, you can get pretty good quality structure from that, actually. And then on the right is a 300 kV microscope that we use for collecting good quality data to really solve structures at good resolution. And so New York actually is very good in terms of the infrastructure that we have. So we have access to 1 of 9 different Krios instruments in the New York City area. That's only part of what's available in New York City. Other universities have their own as well. So it's a very good infrastructure. So what's in the microscope? Do we have to finish in a couple minutes, or?
Yeah, you can take 5 to 10 minutes.
Okay. So essentially, electrons come from the top, come all the way down. The sample sits right here. And then you have different lenses that focus electrons and magnify the image. And then at the very bottom, you have either a camera, in this case, or a detector. So EM has been around for a very long time. And the most critical development that enabled EM being so powerful is this piece of instrument at the bottom, the detector. And that technology development allowed EM to be pushed to resolution that's comparable to X-ray crystallography, 3 angstrom or better. So now you can see actual atoms rather than blobs. So before this, EM, you get resolution about 10 angstrom or so. A good structure by EM is about 10 angstrom. Now you only see blobs. So EM back then was called blobology, blobs. You see blobs. But because of this technological breakthrough, if you will, that enabled us to determine structure. I'll explain how that came about. Okay, so essentially, so samples loaded here, something called the autoloader. Essentially, everything is controlled by computers. Not much human direct intervention anymore. So you can see the basic is a big box. So a magical black box that gets you the structure. So the wavelength of the electrons. So electrons behave as waves. They interact with your sample. The wavelength is about 0.02 or 0.03 angstrom. So much shorter than the X-ray wave we use, which is about 1 angstrom. This is about 0.02 angstrom. Okay, so certainly you can see very much detailed information, but then electrons are very light, so you have to, there are limitations how thick a sample you can go. X-rays are penetrating, go through almost anything. Electrons are very light, so you have to have very thin samples. Anything get thicker, you see black. The electron cannot go through, you don't see it. Okay, so that's the limitation, one of the limitations. So how do we make samples? Maybe I'll stop on this one, then we'll have lunch. So the samples are made, are spotted onto a grid, which is quite small, 3 millimeter in diameter, a grid. How many have done EM here, actually? Okay. So this I borrowed from Yakam. So on the grid is metal grating, and then on the metal grid you have a carbon film which have holes in them. Okay, and the holes is about 1 micron across, and then your sample basically is suspended in the liquid across this hole. And 1 micron is about 10,000 angstroms. So most proteins are about 100 angstrom diameter. Most complex, maybe bigger one, maybe 200 or something. So 10,000 angstroms is certainly big enough for almost anything you want to work with. So you can have molecules suspended in this area, and then you freeze them down. And then, just like cryocrystallography, you freeze them into liquid nitrogen temperature, so it's a vitrified ice. So not crystalline ice, vitrified, so it's glass. And that's the sample you image in the microscope. So if you zoom in a bit more, so each one of these is one of the holes. So you can see one of these square can have thousands of holes in there. And we typically go through only a few squares to collect our data. So, but then you have to find the good squares where your sample is, and then you collect images and you do analysis to get your structure. So the pipeline actually is pretty simple. You purify your protein, you put it onto protein on here, and then freeze it down into your EM grid, and then you take that to screen to see whether your grid has good enough quality in terms of thickness of the ice. If the ice is very thick, you see nothing. You see black. If the ice is very thin, you squeeze all the molecule out of it. Or if you have no ice at all, that's not good either. So Goldilocks, you have to be right in the middle somewhere that's really good. So that's what your screening does, tell you that. And then when you have good enough sample, then you go to the cryos, collect the data set, and see whether you can find your protein or not. And nowadays, just like crystallography, we need a crystal. In Cryo-EM, you need a good sample. Once you have a good sample, you collect data on the cryos, you get the structure within a day or two, or if more complicated, maybe a week. Where the data analysis is very, very powerful, very, very routine nowadays. So I'll show you later today, hands-on. I guess we just did some hands-on now, which seemed to work. So I'll show you some hands-on how to solve a structure using Cryo-EM, go through the process of the Cryo-EM. Okay, but this is sort of the beginning stage. I mean, before this, of course, is protein expression and purification. That we've covered already. Once you have that, you come here. So I guess with that, we'll stop for lunch, and then we'll continue with this after lunch, and then we'll, yeah.